My name is Yuhao and I go by Lauren. I am a graduate student at Emory University and a current fellowship recipient of the International Foundation for Ethical Research. My research is focused on understanding the pathophysiological mechanisms of Alzheimer's disease through studying a tiny but incredibly important region of the brainstem called the locus coeruleus. The locus coeruleus, or LC in short, is among the first region to develop hallmark hyperphosphorylated tau pathology and undergo degeneration in Alzheimer's disease. Its early dysfunction is linked to prodromal clinical symptoms, and its degeneration contributes to later cognitive decline. For decades, Alzheimer's research has predominantly relied on rodent models, but the locus coeruleus in mice simply does not behave the way it does in humans. It fails to show the early LC-first pattern of tau accumulation and atrophy that we observed in human patients. There's also substantial transcriptomic differences between humans and rodents, and as a result, many human-specific disease processes cannot be reproduced in animals, leading to gaps in knowledge and limitation in translatability from bench to bedside. To address this gap, our lab leverages the human brain organoid model. Organoids are generated from induced pluripotent stem cells that retains the donor's genetic background and can faithfully model region-specific characteristics. More recently, complex assembly models that fuses organoids of different regions have made it possible for us to study interregional interactions, which is critical for our understanding of disease progression. Building on this framework, I have developed Tor Knowledge, the first directed protocol to generate human locus coeruleus organoids, which contains fully functional norepinephrine neurons of locus coeruleus that resembles the transcriptomic profiles and neuronal markers of these neurons in vivo. Using long-term cultured locus coeruleus organoids derived directly from Alzheimer's disease patients and assembling them with cortical organoids, we can then model early onset familial Alzheimer's disease and study the mechanisms that make the locus coeruleus one of the earliest and most vulnerable regions of tau pathology accumulation. The goal of my project is to uncover the molecular and cellular bases of regional selective vulnerability and trace locus coeruleus-driven disease progressions and propagations in a human cellular system. To me, this is where the ethical research and scientific rigor intersect. Our organoid system replaces traditional animal studies with a high throughput and physiologically relevant human cellular system, aligning closely with IFER's mission to support innovative approaches to reduce and replace the use of animals in research. Looking ahead, my goal is to continue refining this organoid-based approaches to uncover novel disease mechanisms and facilitate the development of early diagnosis and interventions targeting the early susceptible regions. Ultimately, I hope this work will not only expand our understanding of Alzheimer's disease, but also demonstrate that human-based models can drive breakthroughs while reducing reliance on animal experiments. I want to acknowledge my mentor, Dr. Fikri Bure, whose expertise in human organoid and multi-regional assembly models has been instrumental in shaping this work. I want to also thank the International Foundation for Ethical Research for their support. I am extremely honored to be part of this community, and I look forward to sharing my future progress as my project unfolds. Thank you.